How are you, students? Are you fine? And in this time of pandemic, like Corona, you have to you have to protect yourself by washing your hands, by using some sanitizers, and most recommendably, you should have to stay home. And today we are going to learn about protein synthesis for grade 12 biology students. Protein synthesis, before discussing about the synthesis, we have to discuss about the nature of proteins. Proteins, they make up many living materials. Those living materials are made up of a combination of amino acids. You know, all proteins are made up of single monomers or simple units, which are called amino acids. Amino acids, in nature, there are 20 different types of amino acids. These different types of proteins, which are found in nature, are made up of this combining the 20 types of different types of amino acids. Proteins, as you remember in your grade 11 lesson on unit 2 in biochemical molecules, proteins are biochemical molecules which are formed in ribosomes, or which are manufactured in ribosomes. Let's see some functions of proteins. Proteins have so many or several functions in nature, but among those different functions of proteins, let's see some of the points regarding the functions of proteins. The first one, proteins help fight against disease. The second function of proteins, they build new body tissues, or they are used for repairing of tissues. The third use of proteins is as an enzyme. As you remember in your grade 11, unit 3, enzymes are biochemical catalysts which are made up of proteins. Enzymes are used to digest or for other chemical reactions, and enzymes, they speed up the rate of a chemical reaction. Every chemical reaction in life or in living things are assisted, or there takes place with the presence of enzymes. Enzymes, as you remember, they are specific to heat, they are specific to pH, they are specific to substrates. So these all enzymes in nature are proteins. So proteins are used as a biochemical catalysts. The fourth, the last, but not the least, is they are used as a component of cell membrane. Do you remember in your grade 11, unit 4, cell biology, about functions of parts of the cell and their function? The cell membrane is made up of different components. Among those different components of cell membrane, the first component is phospholipid bilayer, and the second major component of plasma membrane is proteins. There are different types of proteins in plasma membrane. The first one is integral protein. The second one is peripheral proteins. Integral proteins consists of carrier proteins and channel proteins. These are a type of proteins which are globular in shape. And peripheral proteins, some of them are enzymes, and some of them are used to anchor cytoskeletons with carrier proteins. In protein synthesis, the process of protein synthesis follows central dogma. The central dogma for protein synthesis is always transcription is followed by translation in every types of the cells, whether the cell is eukaryotic or prokaryotic. In both type of cells, the central dogma for protein synthesis is transcription is always followed by translation. Let's see these two basic processes for protein synthesis, transcription and translation, one by one. Transcription is a copying of genetic information from DNA into messenger RNA. The reason why DNA is transcribed into messenger RNA is two. The first one, DNA has a genetic material for protein that needs to be made, but proteins are made in ribosomes. DNA is a large molecule. It couldn't leave the nucleus to the cytoplasm because it is very large and double helical structure. But messenger RNA, because of its size is very small and it is single-stranded, so it can leave the nucleus to the cytoplasm for protein synthesis. Because of these two reasons, transcription is very important in protein synthesis. Students, have you got the point? Why DNA is transcribed into messenger RNA? Very good. Let's proceed. The next point that we are going to see is messenger RNA carrying the DNA instructions after transcription, and tRNA, which is transfer RNA, which is found in the cytoplasm, carrying amino acids, meets in the ribosomes, where the actual protein synthesis takes place. Amino acids are joined together to make a protein. This whole process is called central dogma for protein synthesis. Remember, central dogma for protein synthesis, transcription always is followed by translation. As you can see, DNA is copied into messenger RNA. This process is called transcription. And here, messenger RNA, which have a code of DNA during the process of transcription, copies or translates its information into protein. This process is called translation. This is natural rule. Every organism should have to obey this rule. Transcription is always followed by translation. What is a genetic code? Genetic codes are a sequence of bases in the nucleotide that make up code for proteins. As we have discussed before, every protein is made up of amino acids. Every amino acids have their own code on DNA. Then the code on DNA is copied into messenger RNA in the process of transcription, and the codes on mRNA is translated by tRNA to form a protein. This process is called translation. So we have 64 different possible codes for every nitrogen bases in DNA and RNA. In DNA, we have 4 nitrogen bases: adenine, cytosine, thymine, and guanine. And in RNA, we have 4 bases. 3 of the bases are similar with DNA: adenine, cytosine, guanine. But the difference is thymine on DNA is replaced by uracil by mRNA. These 4 different nitrogen bases with 4 different combinations, 4 times 4 times 4, will get 64 possible combinations or possible codes for proteins. But among those 64 possible codes, 3 of them are called stop codes. In the case of messenger RNA, they are called stop codons. These stop codons are sometimes known as translation-terminators. For DNA, there are 3 stop codons: TAA, that means thymine, adenine, thymine, adenine, guanine, thymine, guanine, adenine. These are the 3 stop codes or triplet codes for DNA. When you make their base pairing rule with TAA, UAA, for TG, TAG, UAG, and for TGA for DNA, but UGA for RNA. These are called stop codes. The rest 61 codes are coding for 20 amino acids in nature. 64 possible codes for 20 amino acids. This means one amino acid may be coded by more than 1 codes. Let's take some example. Arginine, it's a type of amino acid which have 6 codes. We'll see it in diagram later. The second type of amino acids that we are going to see is serine. It is an amino acid among those 20 different types of amino acids. Serine is coded by 4 codes. It have 4 different types of codes. The only amino acids which have only 1 code are methionine and tryptophan. Let's see this figure. This one is those 64 different codes for amino acids. For example, serine that we have seen previously have 4 codes: cytosine, thymine, cytosine, thymine, thymine, cytosine, cytosine, thymine, cytosine, adenine, thymine, cytosine, guanine. These are the 4 different codes for serine. So some amino acids have more than 1 code. Some amino acids have 6 codes, like arginine. Let's take valine. Valine is an amino acid which have 4 codes, like GTG, GTA, GTC, and GTT. These 4 codes are coding for valine, and you can study it in your text for further information. So there are 64 different combinations of codes for 20 amino acids in nature. Naturally, we have 20 amino acids only. How does transcription takes place in eukaryotes? You know, eukaryotic cells are cells which have true nucleus. So Transcription takes place in eukaryotes inside the nucleus. And the process is DNA is transcribed into messenger RNA. This is just copying the information of DNA into messenger RNA. And messenger RNA is quite different with DNA. Messenger RNA is very different with DNA. Let's see some differences between messenger RNA and DNA. Messenger RNA is a very small molecule, and DNA is larger molecule. Messenger RNA is a single-stranded molecule, and DNA is a double-stranded molecule. Messenger RNA is very unstable. It is highly mutant. But DNA is very stable molecule. Messenger RNA have uracil in the place of thiamine. That in DNA, the base is thiamine. The sugar found in RNA is ribose sugar, the 5-carbon sugar, which is pentose sugar. And the sugar type is ribose. But in DNA, the sugar is deoxyribose sugar. Deoxyribose sugar. Messenger RNA or RNA, it cannot replicate. It cannot multiply itself. There is no autonomic replication of RNA. But DNA is autonomous. It can replicate itself. It can multiply itself. The next difference is in eukaryotic cells, messenger RNA is found both in cytoplasm and in nucleus. But DNA is only found in nucleus. There are some other differences you can study further. Let's see some steps of transcription in eukaryotes. There are about 5 steps in transcription. The first step is RNA polymerase binds with DNA. Then the transcription factor activates the enzyme. The enzyme begins to unwind the DNA molecule, and it moves along the antisense strand of a DNA, using it as a template, synthesizing the mRNA. The fourth step, polymerase assembles RNA nucleotides. Then mRNA molecule is formed. Finally, the complete process, the complete molecule leaves the nucleus, which is messenger RNA leaves the nucleus. Then the DNA strands rejoin and recoil. This is all about the steps of transcription in eukaryotics. Translation in eukaryotes, as we have said, DNA is found in the nucleus and ribosomes are found in the cytoplasm. This means their site is different. They are found in different sites, in different places. So translation in eukaryotes is found in the cytoplasm, where the ribosomes are found. Ribosomes are found in 2 sites in cytoplasm. The first and major site is rough endoplasmic reticulum. The second site is they are found freely suspended in the cytoplasm. So in these sites, the translation takes place. Ribosomes are found in the cytoplasm. Ribosomes, as you remember in your grade 11 lesson, they are made up of 2 different molecules. The first one is ribosomal RNA, and the second one is protein. Within the ribosome, there are 3 sites 3 sites for protein synthesis: A sites, P sites, and E sites. These 3 sites, A sites, P site, and E sites, are occupied or colonized by tRNA, which is translating RNA or transfer RNA. As you can see in the figure, this is A sites, this is P site, and this is E sites. These 3 sites are sites for protein synthesis. Simultaneous reactions, or consecutive reactions takes place in these 3 sites. Those 3 sites, the whole 3 sites, are occupied by tRNA. tRNA, it is an RNA which polymerizes monomers of proteins into polymers. Let's see some steps of translation in eukaryotes. In eukaryotes, translation follows 6 steps. The first step, the first 2 codons of mRNA enters the ribosome. Then this tRNA with complementary anticodons binds with the first codons that have entered to ribosomes. Then the peptide bond is formed between the first 2 codons of mRNA and the first 2 anticodons of tRNA. There is peptide bond formation. After that, in A sites, complementary anticodon binds with the 3rd codon. A peptide bond is formed between the 2nd and the 3rd amino acids, and the whole process proceeds until stop codons. Stop codons, as you remember before, we have said TAA, TGA, and TAG. These are stop codons. You can say for stop codons for tRNA. So stop codons, they stop or ceases the transcription process. This is all about translation in eukaryotes. So, is there any difference between transcription and translation processes in eukaryotes and prokaryotes, students? Yes, this is obvious because prokaryotic cells doesn't have nucleus, and the site of protein synthesis, both processes, transcription and translation, takes place in the same sites, which is in the cytosol, because there is no different sites for transcription and translation. But for eukaryotes, there is a separate sites because DNA is found only in the nucleus and RNA or ribosomes are found in the cytoplasm. Because of different in sites, the processes takes place separately. Let's see. Unlike eukaryotes, prokaryotes do not have nucleus. Because they do not have nucleus, there is no post-transcriptional process in prokaryotes. Post-transcriptional process means modification of mRNA before translation process. In this case, eukaryotic cells undergo post-transcriptional process, which is modification of mRNA. What do we mean by modification? Modification of mRNA means after the transcriptional process, the resulting molecule is RNA with introns. Introns are wastes, which is outside or on the opposite sides of the mRNA. They should have to be cleaved. They should have to be cut before leaving the nucleus. This process is called modification. This modification process is only takes place in eukaryotic cells. This is called what you call it post-transcriptional process. But post-transcriptional process in prokaryotes is not occurring. Transcription and translation processes are coupled in prokaryotic cells because there is no different site for DNA and ribosomes. They both are found in cytosol of prokaryotic cells. That means when we mean the processes are coupled, both processes, transcription and translations, takes place simultaneously. During the transcription process, translation is also followed the process because the site is in the same site. The next thing that we are going to see is transcription and translation are separate in eukaryotes. Eukaryotic transcription takes place in the nucleus, and eukaryotic translation takes place in the cytoplasm. Eukaryotic mRNA is modified before leaving the nucleus. That means introns are removed or cleaved from mRNA. And finally, what controls gene expression? You know, every body cell, let's take human body cell, have 46 chromosomes. Every cells, blood cell, nerve cell, muscle cell, cardiac cell, and so on. Okay, students, what controls gene expression? Every cells of the organisms, let's take human cells, have every traits or genes for every characteristics. For example, blood cell, muscle cell, cardiac cell, nerve cell, and so on. These all types of cells have all genetic information of the organism. But every cell, because they are specialized for a specific function, they have their own types of protein secretions. So, all cells are having every traits, but all cells are not producing proteins for a specific site. This is controlled by gene switching off and gene switching on. In every cells, there are some genes switched off and there are some genes switched on. Gene switching on is controlled or done by transcription factors or activators. If the cell needs a specific type of protein, a gene which is responsible to secrete that type of gene will be switched on, and it will copy its information into messenger RNA, and messenger RNA translates it into protein. But in that specific cell or in that specific site, the type of protein which is not needed is switched off. The gene which is responsible to order the secretion of protein is switched off, and the cell would no longer synthesize a protein, that specific type of protein. This is controlled by siRNA. siRNA means short interfering RNA. This type of RNA switches off some type of genes in order not to secrete a specific type of protein. For example, glands in our body, there are different types of glands. Some glands secrete enzymes, some glands secrete hormones. Enzymes are proteins and hormones are also proteins. But glands which secrete hormones would no longer secrete enzymes. And glands which secrete enzymes would no longer secrete hormones. Why? Because glands which are secreting enzymes, they switch on the gene of enzyme secretion, and they switch off the secretion of hormones. By this way, different cells in our body have their own specific function. siRNA, they silence gene expression, either by stopping copying of information or by cleaving or cutting mRNA, messenger RNA, not to translate the codes of protein to protein molecule. Is it clear, students? Today, we have learned about protein synthesis. Protein synthesis, when we start our lesson, we have seen the nature of protein, the function of protein, the central dogma for protein synthesis, which is transcription followed by translation. We have seen what transcription means and what translation means. And we have seen transcription in eukaryotes and translation in eukaryotes, and the difference between transcription and translation between eukaryotes and prokaryotes. And also we have seen each steps of transcription and translation in eukaryotes. Finally, we have seen gene expression. Thank you, students. Stay safe, stay home.